Hey guys, so first thing, I am not at home because my internet has not been working since the last Wednesday, but I wanted to go ahead and record this so that you will be able to watch it this week. If I get a chance, I will redo it. Hopefully it's not super loud, but there is a little bit of music playing in the background. Okay, so for Unit 5, cellular respiration, we have talked about the purpose of cellular respiration really is to make ATP for our bodies, but it's gonna do that through glucose. So as we intake glucose, our body is gonna break down that glucose and turn it into ATP, and we will talk about how that happens later on. But know that the main purpose of cellular respiration is to break down glucose and turn it into energy that your body can use to do everything, from breathing to your heart beating to you being able to exercise and all of that. Okay, so who uses this? All living organisms use cellular respiration, and there are two types. We have anaerobic respiration, which is going to not use oxygen. So or ana, an is going to mean no, so no aerobic. And aerobic is going to mean oxygen, so not using oxygen. And this is going to be really inefficient. So your body is not going to make many ATP by using this type of respiration. Then we have aerobic, so just oxygen, which basically means it uses oxygen. And that is going to be super efficient. We'll find out how efficient later on, but definitely much, much, much more efficient than anaerobic. So we need to make sure we know these two types and the differences between them. We're going to start with anaerobic respiration, and we're going to go through all of the steps of that. The good news is they both start with the same step. So anaerobic and aerobic are going to start with this step here, glycolysis. And glycolysis, I've preached a little bit about prefixes and suffixes the whole semester, but it's really important that you know these things because in the future it helps you figure out other words that you may not know the definitions of. So glyco means sugar and lysis means to break apart, and that's exactly what's happening during glycolysis. Sugar is being broken apart. It breaks down glucose to obtain ATP molecules. All right, so this occurs in the cytoplasm for anaerobic and aerobic. This is the first step, and it occurs in the cytoplasm for both of those. So yeah, first step in both aerobic and anaerobic respirations, and we have an enzyme here. An enzyme is going to be something that lowers that energy that it needs to make the reaction happen, and these are going to be super important in our whole process here to end up making a lot of ATP for us. So that's NAD, it's going to pick up H's to make NADH. So what happens during glycolysis? Two ATPs are used to break apart glucose. So it's going to need two ATPs to already exist in the area, in the cell, wherever, that it can use to go ahead and start this process. So after we use our two ATPs, it's going to break apart glucose, and we're going to get two pyruvic acids. And we have this whole little thing here. And I really want you to be able to understand the difference between net and gross energy. So the process actually makes 4 ATPs, which you can see here, the 4 ATPs. But we already had to use 2, right? So we had to use 2 ATPs, which is going to really leave us with 2 new ATPs. So 4 came from how many were made, minus the 2 that we had to use, and now we have 2 ATPs net. So our gross number is 4, and our net number is 2. All right. So products of glycolysis. So from the breakdown of glucose, that's that molecule right there, the C6H12O6, that is glucose. And it's going to break that down and make those 2 pyruvic acids, 2 ATPs net, and 2 NADHs. So those NADH are going to act like a taxi cab, or an Uber would be something that you're more familiar with probably, that's going to transport those H+ ions. And that's going to be really important during aerobic respiration, but we still have those in anaerobic. So if there is oxygen available after all of this takes place, it's going to go and move into aerobic respiration. And aerobic is going to be the one that uses oxygen. If there's no oxygen available, then anaerobic respiration is going to take place. And so in our case here, we're going to go ahead and move into anaerobic respiration. So anaerobic, no oxygen is available, but glycolysis is still going to take place. And then we're going to move into fermentation. So fermentation is the second step of anaerobic respiration, and this is also going to occur in the cytoplasm. So glycolysis is in the cytoplasm and fermentation is in the cytoplasm, which means all of anaerobic respiration takes place in the cytoplasm. We have two types of fermentation, that's alcoholic and lactic acid, and 0 ATPs are made throughout the whole fermentation process. So either one, alcoholic or lactic acid, is still going to be 0 ATPs made. Who uses anaerobic respiration? We have bacteria, plants, fungus, and animals, but only in their muscle cells. And we'll talk about that a little bit later on. So let's go through those two types. We'll start with alcoholic. This is going to be used in yeast, plants, and bacteria. Again, 0 ATP are made, and this is going to be our formula here. Both of these fermentation processes are going to start with the same two reactants. So we're going to have the 2 pyruvic acids, which came from glycolysis, and the 2 NADH, which also came from glycolysis. This is super easy to remember because alcoholic fermentation, you're going to get 2 alcohols, right? So alcoholic fermentation produces 2 alcohols, 2 carbon dioxides, and 2 NAD. So we start with 2 and 2, and we get 2, 2, and 2, right? So 2 is our number for this. And we shouldn't have any trouble remembering because alcohol produces alcohol. It's also going to be used to create food and beverage products like alcohol, right? So we get our 2 ethanols from alcoholic fermentation, and it's going to be used to create beer, wine, spirits, and bread. So make sure we know these products here, and make sure we know some foods that are created using alcoholic fermentation. Then we have lactic acid fermentation. That's our other type. And we're going to use this, or fungus, plants, bacteria, and animal muscle cells are going to use this. So again, all fermentation, 0 ATP are made. And we start with our same reactants. So we have 2 pyruvic acids and 2 NADH. And it's going to yield the 2 lactic acid and 2 NAD. Just like alcoholic, the type of fermentation actually yields that product, right? So we have lactic acid fermentation. It's going to make lactic acid. Lactic acid is actually the stuff that builds up in your muscles that causes you to be sore. So you've ever heard of like break up the lactic acid? That's what they're talking about. So when your body doesn't have enough oxygen in its muscles, lactic acid, it has the chance to form, and it causes you to be sore. So some foods, we had our 2 products there, 2 NAD, 2 lactic acids, and our food products made from this process are yogurt, soy sauce, sauerkraut, and kimchi. So I would know some of those. And then a review. So it all occurs in the cytoplasm. We just have 2 steps. We have glycolysis, 2 ATPs, fermentation, either type makes 0 ATPs. So it's super inefficient, right? We only get 2 ATPs per glucose, which is not very many. So anaerobic respiration, no oxygen. It all occurs in the cytoplasm. It's got 2 steps, and it makes 2 ATPs. All right, then we have aerobic respiration. This is our formula for that. We're going to put in a glucose. Remember that formula is glucose, that molecule, plus oxygen equals carbon dioxide and water. So we're going to go through this step, but it's used by all eukaryotes. So we talked about prokaryotes and eukaryotes. You are a eukaryote, if that brings back some differences between the two, but it's going to be used by all eukaryotes. So aerobic respiration, it requires oxygen to make ATP, and this process is going to start in the cytoplasm with glycolysis, and it's going to end in the mitochondria, which has 2 other steps, and those 2 steps are going to take place in the mitochondria. It's going to make 18 times more ATP than anaerobic respiration. So remember how I said anaerobic was super inefficient because it only made 2? Well, aerobic is going to make 36 ATP throughout all the steps. So 18 times more, super efficient compared to anaerobic. Okay, so the structure of a mitochondria, it has an inner and outer membrane, which you can see here, and then the cristae and the matrix, which are going to be the places that the last 2 steps of aerobic respiration are going to take place. So 3 steps of aerobic, we have glycolysis in the cytoplasm, Krebs cycle in the matrix, and electron transport with oxidative phosphorylation in the cristae. So glycolysis, we've already been over this, occurs in the cytoplasm, and these are the products, 2 pyruvate or pyruvic acids, 2 ATPs, net. Remember what net means. We used 2, it produced 4, so we only have 2 left that are new, and then the 2 NADH. We have this transition phase in aerobic respiration where this pyruvate from glycolysis is going to match or meet up with this enzyme, and it's going to produce acetyl-CoA. So acetyl-CoA is what we need to move into the Krebs cycle. So this is going to take place in the matrix of the mitochondria, and the Krebs cycle needs citric acid to begin. So acetyl-CoA is going to meet and bond with oxaloacetate, and that is going to end up creating citric acid. Some products that occur during this whole process are 2 ATP, 6 CO2s, which are carbon dioxides, and you can follow this as it goes around the cycle. It's just going to be continuous, right? As we put in new new citric acid, it's going to keep doing this. We're going to get 2 ATPs, 6 carbon dioxides, 8 NADH, and 2 FADH. Again, remember, these are going to help transport those H pluses into the next step. So then we have our electron transport chain. This occurs in the cristae of the mitochondria. And those are super easy questions if you see them on a test to get right. So make sure those are things that you really remember. And then the NADH and the FADH2 are going to enter and create a H plus gradient, which means they're going to be more H pluses on one side than there are on the other side of the membrane, right? So we're going to get some other products in this, and we're going to get 6 waters. And then the NAD, once it releases its H plus, is going to go back to glycolysis, so it can do it again. And FAD is going to return to the beginning of the Krebs cycle, so it can help again, right? And then, so here's our electron transport chain. You see all the H's. NAD drops off its H's here at this first complex. FADH is going to drop off its H's at this second complex, and the third complex is going to send them through. We get, here's our water, you can see. And then this one right here, this complex is going to be the most important. This is ATP synthase. So ATP synthase, since we have this concentration gradient and there's going to be so many more H's up there than there are down here, the H's want to get back over, right? Because H's, they want to go from a high concentration to a low concentration. Just like if you're in a crowd, you might want to get out and go to somewhere where it's less crowded. So as they flow through the ATP synthase, it's going to put energy in, and we are going to end up having ADPs in there, and another phosphate is going to be attached to the ADP as it flows through. So ADP plus another P is going to equal ATP, right? Because the only difference here is that ADP has 2 phosphates and ATP has 3 phosphates. So this is going to have these 2 paired together, and now we're going to have ATP. So as these H's go through, we're going to create ATP. And this is going to be called chemiosmosis. So a concentration gradient causes H plus ions to move through the membrane, just like we talked about. So H pluses are going to move through here through the ATP synthase, and as it creates, it's going to create ATP as it does that. And we're going to end up with around 32 ATP. So this last process, this electron transport chain, is going to end up creating 32 ATP. And this is the picture is just showing you, we start with glucose, it's broken down into pyruvate. During this little transition, we're gonna end up getting citric acid. It'll enter the Krebs cycle, and then we'll move through the electron transport chain. And this shows you how many ATPs are created in each step. And here's our mass format. So glycolysis is 2, Krebs is 2, electron transport chain is 32. And so that equals 36 ATP per glucose, much more efficient than we saw with anaerobic respiration. Another picture, kind of all the same things, just in a different way. Start with glucose going into glycolysis, broken down, we get ATP over here, and then all of these carry on the hydrogens into the Krebs cycle with our 2 pyruvates, and we get 2 ATP out of that. And then as the enzymes take the H's and drop them off, we're going to end up getting our 32 ATP down there. So feel free to ask me any questions, email me if you need too. I hope there's not too much background noise in this video. I didn't really have another option. So, yep, if you have any questions, just let me know. Thanks.